It's my pleasure to introduce today's speaker. His name is Gebrehaweria K and he came from Ethiopia. Reda is a PhD student at the Doctoral School of Animal Science. He is supervised by Előadó and co-supervised by Levent and Szegedi from the Agricultural Campus. Reda is doing his research on the nutrient sensing mTOR pathway, and he's working with Japanese quails. And he's in his final year, so today he will talk about his most interesting results. So you can start your presentation.
Okay, thank you very much, everybody. Thank you. It's my pleasure to be here to present my research output that we have done in the last 3 years with my supervisors and my colleagues. I'm just supervised by Professor Adam and Professor Levente from Animal Science Department. I will be presenting part of our results. Let me start with some brainstorming questions that will lead us into the results. We know that nutrition is important for growth and development and for reproduction and so on, for immunity, but scientists are just struggling to know how are in detail these nutritions are working. What is the mechanism behind nutrition is affecting our life or the life of organisms. We can also see how do organisms allocate resources among stritrators. So we know that resources are scarce and organisms has to decide to allocate into different life history traits depending on the availability, depending on the amount and the quality of resources. So this is also one important study in this contemporary science. Who are the main candidates of nutrient sensing pathway? Who are receiving, who are sensing the nutrients in the environment? And this one is also very important. That's why it takes us into the core of the nutrient sensing pathway, or the central unit of the nutrient sensing pathway. The other one is which genes are upregulated or downregulated because of nutrition. When it is high amount of nutrition, when it's low amount of nutrition, some genes has to be expressed and some genes has to be, in response, has to be downregulated. So this will be how organisms manage their growth and reproduction and immunity and lifespan. Do genes expression mediate resource allocation? So can we find a relationship between gene expression, especially the steady state gene expression, and the resource allocation among the life history traits? Does genes show specific nutrient, response to nutritional availability? So do males and females show different response to the available resource, to the resource that they find, or to the condition, the nutritional condition? So we will try to touch some of them, but we will not try to answer these questions because these are really big questions. So in a very broad sense, nutrition is provides a building block of this. We know just these are the building blocks of the nutritional content, such as the glucose, amino acid, and fatty acids. These are very critical for the cellular metabolism. So we know this one, everybody knows from our in primary schools. So it also influences gene expression or gene transcription. Which of them are expressed or downregulated are just the previous questions. That will be our interest. It also impacts the epigenetic modification, that is heritable gene expression. We know there is a steady state gene expression. There is also heritable gene expression, which is most of the time called epigenetic modification, where it is transferred into from parents to the offspring. So especially nutrients like methionine are very critical in epigenetic modifications. So these nutrients are important for this one. It modulates signaling molecules, especially the insulin and insulin-like growth factors. Signaling is the most important thing, so that is modulated by the nutrition to regulate the cellular process at the whole. It regulates antioxidant defense mechanisms to mitigate oxidative stresses. Yeah, these nutrients are also important on the antioxidant defenses mechanisms. So nutrients are the source of oxidative stresses, and also they are also the source of mitigation, or they are also the solution for oxidative stresses. So this is generally how nutrients are working in our body or in the body of organisms. But when we can squeeze our questions into our point, we can see resource allocation, how our organisms allocate resources when we know that there is certain condition, so organisms has to fit into the condition. We know this one many times, or you may heard a lot of presentations about this. Resources are scarce, yeah, and organisms allocate, especially this in the outside environment. Resources are scarce always, and organisms has to allocate it to the competing traits, especially this growth, reproduction, and somatic maintenance, defense, that is the immunity. So they have to balance the resources to give into one of the traits, or to both of the traits, or to all of the traits. So this is a very important thing to know. So during the early stage, during the early life, organisms most of the time allocate most of the resources for growth and development. This will help them to increase their survival, their lifespan, when they have a good nutrition during the growth stage, at the growth stage. And also it determines the reproductive lifespan, how much they will reproduce, and the quality of the product, I mean the offspring will also be determined by the resource they found and allocate at the early stage of their growth. So it's very important to maximize their survival and future reproduction success, including the quality and quantity. But once the organisms get matured, they will be tending to allocate most of the resources for reproduction if there is enough amount of resource or nutrition. So they will allocate resources to reproduction and somatic maintenance, depending on the availability. If there is high amount of resource, moderate amount of resource, or just a low amount of resource, they will decide to allocate into this one. So it depends. Organisms allocate resources depending on their stage of growth towards different life history traits. Sorry. So here we can see kind of where different scientists say different things about this one, but we put this graph, like for example, under certain condition, organisms may put a lot of resource to their somatic maintenance than to the reproduction, and at a certain stage, they may put equally to the somatic maintenance and reproduction. Okay, this may be unlikely to happen, or it happens many times. This one indicates where organisms are resembling for reproduction. They put a lot of resource for reproduction while compromising this one, or just keeping this one. So it depends, especially it also depends on the species. Some species may put their effort on reproduction at once, and they put regardless of their somatic maintenance, regardless of what happened to their life, they will put all of their resources or most of their resources into reproduction, because they will reproduce in the early stage or at once or 2 times, and they will die. So they maximize their reproductive efforts. Some of the organisms will allocate into somatic maintenance because they need to stay long and they have to reproduce a lot, or still whether they cannot reproduce but they have to stay long. So the organisms, depending on the species, and depending on the availability of the resources, can decide where to allocate their resources. So we will see a little bit in our results too. So these are questions related to this one. At what condition is this to be happen? Is it as the excess resource? Considering, for example, Japanese quail as a model organism or as a research organism, what happened at the excess resource or at a moderately less resource, moderately less, not just extremely less, but what happened at the extremely less resource, limited resource, and what happened to the organisms when availability of resources unpredictable. Just today is a lot, and tomorrow there will not be any food, and then how could organisms harmonize themselves into this kind of conditions? So this graph will be different depending on this one. Studies have been studied, said something, but it depends on the species they study. So to highlight about the crucial nutrient sensing pathway which leads us into our target genus, nutrient sensing is a mechanism by which cells are sensing into nutrient available nutrients, and they respond accordingly. So this is a very critical pathway among the whole pathway, among of the maybe a lot of to call in numbers the pathways. So the nutrient sensing pathway is very important. So the mechanism involves a variety of cellular receptors and signaling pathways. The main ones are the mechanistic target of rapamycin, which is the mTOR pathway, which is crucial that we are going to focus today, and the AMPK, which is adenosine monophosphate activated protein kinase, which is always the sensor of the availability, energy availability in our body. We will not talk about this more, we will focus on this one. So we can see this mTOR is very critical. It senses a lot of incoming chaoses, and it responds to this one. It will give this one. We will see how it will work. So the coordination of resource allocation is proposed to be attributed to a key genes in the mTOR pathway. So the genes in the mTOR pathway are mostly responsible for the availability of resources. I'm sorry. So the interplay among the mTOR genes, pathway genes, coordinates anabolic and catabolic process in response to the growth factor and nutrients. So the nutrients, this is a very critical central unit in the nutrient sensing pathway. So I will show you just in short, just as a click, the interplay, the mechanism, or the interplay of the genes. So here we can see the IGF-1, the insulin like growth factor 1, which is most importantly produced in liver, and it works as a growth factor and in response to growth hormone and nutrition. Nutrition and growth hormones, maybe insulin also initiates the activity of this one or the production of this one, this IGF-1, and this will take us into what will happen into mTOR, the mechanistic target of rapamycin, which is the central unit in the, they also say that it is the protein machinery inside the cell. So when nutrition is available, so it will initiate the production of high-level IGF-1. This will send a signal into the cell through its receptors, and the signaling cascade will give a signal to the mTOR, which will tell us that there will be something outside the cell, which is initiated by the IGF-1. And this nutrition can also send amino acid, especially these essential amino acid, mainly leucine, methionine, and some lysine, will trigger the activation of this one, the activation of mechanistic target of rapamycin. So nutrition affects in two ways. One is through the amino acid influx into the cell due to the amino acid carriers or transporters. We will have either less or high amount of amino acid in the cell, and this will trigger the activation of this kinase. It is a kinase protein. While this one also gives a signaling cascade. Those both signals will give this to be active. So it senses the amino acid. It also receives this signal from the growth hormone or the growth factors. So while maybe due to shortage of food or less resource, the other MPK pathway will inhibit this one. This is during the shortage of food. So the mTOR will be downregulated because of this pathway. But we will not just focus more about this one. We will shortly just see this one. So in response to the activation of mechanistic target of rapamycin, the ribosomal protein kinase, this is the translation factor, which is very important for protein translation at the lysosome, at the ribosome. So this ribosomal protein kinase will initiate the translation of proteins, and protein synthesis will be activated, or just a lot of proteins will be synthesized. And also this, for ABP, that is eukaryotic initiation factor binding protein 1, this is also critical in, if this is active, this will be inhibited. The initiation, the eukaryotic binding protein 1 will be inhibited, because if this one is active, it will inhibit the activity of the protein synthesis, because it will attach the eukaryotic initiation factor 4, and then this will be inhibiting protein synthesis. But when this is active, it immediately phosphorylates this one, and this one will be inhibited. So it will make this to be much more active, and protein synthesis will be more at high level. So this will give us for growth and reproduction. It will the protein synthesis will initiate cell growth, cell reproduction, differentiation, and so on. It will give into aging, and then this will take us into. So when this is active, also it will downregulate the autophagy pathway. When this is active, this one will be downregulated. This one will be activated. This pathway will be activated, and then. But this autophagy pathway will be activated when this one is downregulated, but when this one is, when the FOXO and AMPK are activated because of maybe less amount of resources. So this will inhibit the growth and reproduction plus the aging. So they will extend life of organisms. So they are expected to increase the life of organisms because of reducing the oxidative stresses inside the stress and the cells and the tissues. So this is our question: what happened to nutritional limitation that we studied, and what happened to unpredictability when food is unpredictable, fluctuating across time, or in days, or in weeks, or in months. So. This is our question. So why we are focusing on gene expression, I will just give very highlight So gene expression involves, we know that it involves from translation and transcription up to the protein synthesis, but there are a lot of processes inside this one. The transcription, translation, and there is also a turnover of mRNA and protein. We know that there is a short half-life of mRNAs and proteins. So when gene expression, from mRNA expression up to the phenotypic expression, there are a lot of activities happen. But still, the mRNA expression or the gene expression, we call from now gene expression in the mRNA expression is still important, while people are still recommending to study that. So, for example, these guys studied these mRNA abundance is correlated, highly correlated, or just not highly, but it's correlated with this protein abundance. So if we study this one, just it's a proxy for this one and for the protein availability and for the even for the phenotype, because proteins are near to the phenotype. So we can also study with this. This guys also studied the the mRNA expression is extremely highly correlated with the protein. So it's important to study gene expression at the steady state gene expression. While studying proteins is very sophisticated, and studying mRNA is relatively lower in sophistication. It's more easier than studying the protein. So we can still study the mRNA expression as a proxy for studies this gene expression totally. So what is the importance of this mTOR into the genes expression? I'm not going to talk about the importance of gene expression because we talked it already, but what happened to the gene expression? So we can see after, as I talk, after it gets a signal from this one, it will be activated, and it will it will activate these transcription factors like this one, this one, and this one, and the activation of this one will give different, will do different activities. For example, when this one is activated, it will not go to the nucleus and it will not activate this. For example, when this one is activated, this is a transcription factor for autophagogenesis, so it will go outside. It will not go to the nucleus for transcription. But when this one is activated by the mechanistic target, it will go into the nucleus. It will be transported into the nucleus, and it will just facilitate the transcription. So mTOR is still important for gene expression, not only for translation, but for transcription also. By itself also, it transported into the activated protein. mTOR can also transport it into the nucleus. It can also facilitate transcription by itself also. It's a transcription factor by itself, while also a lot of transcription factors are affected by this one. Some of them are hindered from transported into nucleus. Some of them are also initiated. So we can see when this one is downregulated, this one will not be facilitated. Phosphorylated, and it will go and it initiates this one. But what happened during the restriction and unpredictability? That's our question still. So this is our study. We conducted 2 consecutive experiments. The first one will be answers how quantitative data restriction affects gene expression and the phenotype. How are these gene expressions affect these 2 important linked stuff? How birds allocate resources between body mass and egg production. That will be our key findings in this study. So we had 20%, 30%, and 40% restriction. That is quantitative feed restriction. So we have female and male quails that accounts for about 8. So it was an individual study, individual bird study. We manage birds at an individual level. We don't manage them in group. So that will make it very important because it was highly controlled. So we find that body mass is, it's obvious that during restriction, body mass is affected in both males and females. You can see, but most of the time, it is highly pronounced in the females. In the very beginning, their body mass is higher, while the effect is more also pronounced. You can see even between the restrictions there is a difference, but here you can see, for example, the 30% and the 40%, the 20% has no that much difference, maybe because of the start, but the 40% still go down because this is a most severe data restriction. So this indicates that our restriction experiment was working. So what happened to the egg production? On the egg production, you can see the probability of egg laying, daily egg laying, and the total egg numbers that has been, our experiment is conducted for 14 days. So these are the eggs laid on the 14 days. So statistically, only the 40% data restriction showed a less number of eggs than the others, even though they show the tendency. Of course, some birds will skip laying in all of the restricted groups, but this one is statistically different. We can also see the probability of laying is very shallow in the 40%, while, okay, it's affected here also, but it's not this much. The reason why it is, this one can also be expected that the starting point was still low because of different reasons, but still the 40% is statistically lower in egg production. What about the egg number? Egg mass. Egg weight is, you can see it's less under the 30% and 40% restriction. Okay, so we can think that, okay, on the egg number, we can see that 30% was not different from this one, but here the 30% and 40% are different from the control one. This is the ad libitum fed group. So the 20% is almost not different from this one. While you can see on the body mass is reduced in both of them. In all of them. Okay, what happened to the gene expression? In this study, we analyzed this 6 or 7 genes, I think. 7 genes. The growth hormone receptor, IGF-1 receptor, the mechanistic target of apomycin, the ribosomal protein kinase, and autophagy, 2 autophagy genes. We analyze these genes because they are found under the same pathway, cascading pathways. So when we come into the results of this gene expression, we showed that in females, the growth hormone receptor was increasing. That was surprising for us. We were expecting that growth hormones will be reduced because of data restriction, but suddenly it is increased. We can talk about this, about birds, what happened in other organisms. The result is different, but here, in both the males and females, it shows on the severely restricted groups, growth hormone receptor is increasing. Of course, we didn't find previously studied results, but I don't know. Okay, what we call the Bird's paradox and so on may come here. So this one is upregulated. IGF-1 in response, while we are expecting this one to be increased because of this increasing IGF-1, growth hormone receptor, but this one is downregulated. Of course, IGF-1 is not only regulated by growth hormone, but also it's regulated by insulin. But we know that birds are insulin resistant, so we don't know if insulin is working on behalf of growth hormone here. So it is downregulated. Okay, but we were expecting this one and this one will be upregulated together or downregulated together. But if in response to dietary restriction, they have to be downregulated. So we also see this growth hormone receptor. I mean the IGF-1 receptor. This IGF-1 receptor was almost similar. We found it as we expected. So it is less at the restricted groups, especially in the males, but in females there was no statistical difference, but yeah, we show some trends of reduction. Well, these are also the mechanistic target of rapamycin. Respond to here, see two genes. This one you can see in response to the dietary restriction, we see even it is reduced with the severity of the restriction, while in males all of the restricted groups are showing the same results. So the severity here is high, okay, maybe because of egg laying. They are showing less, I mean just severe effect on relative mTOR expression. We can also see the ribosomal protein kinase. This was also surprising us. We were expecting to decrease because of dietary restriction, but it's increasing. And surprisingly, it's with both of the sexes. So in other studies, especially in mammals, in mice, it was reduced in response to dietary restriction, but here it's increasing. We cannot corroborate it with a study, with other studies, but it needs another, further study. How are transcription factors working against this one and so on in birds? We can also see the autophagy genus. The autophagy genus showed increasing trend in both of the sexes, but in males, this autophagy gene 9 was increased at all of the restriction levels, but in the females, only the 40% was restricted. Was higher, significantly higher, increasing. So we say that this autophagy genes are increased or expressed in response to downregulation of the mTOR. When mTOR is downregulated, this autophagy genes has to increase because always mTOR is against this one. Here we also see this autophagy gene 5 to support to this one. Yeah, we see the trend, but mostly only in females is significantly higher in the restricted groups. But here there is a trend, but not really at a statistically significant level. So these are the autophagy, what happened to the autophagy genus, and this is what happened to the autophagy, to the gene expression in response to dietary restrictions. Finally, we try to see in this experiment, we try to see how are this dietary restriction affects resource allocation. So we designed this illustration. What is the imagination? The imagination is, for example, there can be a resource allocated to the reproduction at a certain condition, and there can be a resource allocation to the somatic maintenance at a certain condition. So at what condition are they able to do this? Will be, let's see our actual, this is our actual data. So we see that there can be a resemblance or a decision to allocate to the reproduction or to the somatic maintenance here. This is the ad libitum, and both of the proportion to the body mass and to the egg mass are increasing here. But in the 20% restriction, birds are, even in the 30%, birds are resembling to allocate resources into the reproduction than into the somatic maintenance, to the body mass. So they put their resources into the reproduction because this is a proportion of egg mass to change to the initial body mass, I mean to the initial egg mass. While the 40% shows a resemblance to allocate more resources into body mass or into somatic maintenance. We can say it, okay. In this condition, there is a body mass lost, but the egg weight was tried to maintain, especially in the 20%. But in the 40%, all the egg mass, egg number, and body mass was totally reduced altogether. But here, birds may decide to put the resources into their body mass than into the egg mass. So we found this one. So it was interesting. We also tried to see how gene expression are affecting this process, the process of resource allocation. And we see only the mTOR was having significant relationship. So individuals with lower mTOR expression this was also really surprising. So birds having low expression was tending to allocate more resources into reproduction than birds having high expression, high mTOR expression. So those birds who have higher mTOR expression are tending to allocate more resources into body mass. So those having less mTOR expression are tending to allocate more resources into the reproduction. So but with the other genes, we didn't find a relationship. Let's see a highlight of these sex-specific responses. Yeah, we know that Japanese quails are size dimorphic. So the female are giant in body mass, while males are low. I mean, just smaller, not just small, but smaller than the females, but a little bit aggressive. So in response to data restriction, so you can see the biomass, the body mass in the ad libitum group, they are higher. But here, in response to data restriction, okay, the females show high level of reduction here. And also, we can see also this one. Finally, at the week 2 and at week 1 and week 2 in the 40% and 30% restriction levels, their body mass becomes almost equal. That means males were not that much reducing a lot, but females were just reducing in every step. So here, the level of decreasing in body mass was higher in females. So that is a sex-specific response to data restriction. Yeah, it's obvious that because females are laying eggs, so they have to use a lot of resource to lay their eggs, and they have to manipulate, or they should not just use all the resources for their body mass. So this is a sex-specific difference in gene expression. We see this one in mTOR. Females showed at the 30% and 40%, it's highly reducing, and even finally it becomes almost equal, statistically equal with the males. Here, the ribosomal protein kinase has increased at the 20% and 30%, but we didn't really justify this one. And we also saw that this catabolic gene also showed higher on the males at the 20% and 30% restriction levels. So there is something that shows a similarity with this one, but we expect they are totally different, but now they are showing almost similar way of expression. That was our first experiment. So going to the second experiment, let me show you not much in detail, but some of them. So in this experiment, we tried to answer how do organisms respond to nutrition, nutritional daily unpredictability. So do males and females respond differently? So this was our design. The we give the control group was eating 100% of their intake. We had another group that is similar to what we have been done in the first experiment, that is 40% restriction, but restricting constantly throughout the 2 weeks. But the other one is just fluctuating the food across the days. So you can see in this case, we give them less amount of food, that is 40%, 6%. Here we give them 170, which is ample amount of 30. We also reduce into this one, and then this, and just you can see how the food is fluctuated across the days. So we try to see that do birds respond to this kind of fluctuations, daily fluctuations in food availability, or they just escape, okay, there is no food tomorrow, or otherwise I will find another food yesterday, and so on. So we found that in females, okay, both, especially we will focus in this discussion, we will focus on the unpredictable one. The unpredictable one showed still less amount of, less body mass. That is, it reduced. But in males, it doesn't show anything. So males don't care about the fluctuation in food availability. But in females, they were serious in body mass still. In number of eggs, you can see that daily unpredictability and control, they are almost the same. So they don't show any kind of, while they respond into body mass here, they don't respond to egg number. So they might have compromised their body mass, but they don't do anything on their, they didn't respond to the daily unpredictability. Here, in egg mass also, it was similar. They didn't respond to daily dietary unpredictability. You can see in the daily, here it was, in the 40% it was reduced a lot, but in the total body mass also, you can see this one is reduced, this one is not. We analyze some genes, we add some 3 genes, I think, or 2 genes into the other genes that we analyzed in the previous experiment. So we found that IGF-1 showed reduced but statistically not different at the unpredictable one. Similar in males, but statistically not really significant. Here is what happened to the IGF-1 receptor. The IGF-1 receptor showed something which is different here, but at the unpredictable level, it doesn't show anything with unpredictability. This one is the FOXO1, one of the upstream of mTOR. This is one of the major transcription factors, important for downregulation of mTOR and upregulation of this autophagy. So it showed that, okay, there is an increase in this, but statistically it's not different. Here it's almost the same, but still there is a trend to increase. What happened to mTOR? To mTOR, both of them are increased, decreased in the 40% and in the unpredictable one. It was really interesting, while the others are not showing any kind of interest. Reduction here, it shows reduction. In males, it doesn't show, but it seems like low in the 40% restriction. Okay, the ribosomal protein kinase, still it shows similar, consistent trend in the males, but in females, the unpredictable one is higher than the other one, especially the control one. Still, for us, it's hard to explain it. We need to go in detail to study about this gene still. while we expect to go in harmony with the mTOR, but it's going always against the, this is a gene, but of course, at protein level, it works differently, because when there is high amount of activated mTOR, ribosomal protein kinase will be activated and it will initiate the translation. But at a gene level, it's different. So it needs some explanation. There is also this eukaryotic initiation factor, binding protein, which is downstream effector of this mTOR. Statistically, it was not significant, especially with unpredictable one, but here in the males, it showed a lower, which is still also against our understanding. These are the autophagy genes. We try to see the ALK1, which is direct downstream of mTOR. It showed that the unpredictable shows higher ALK1 expression, but in males still it's not that much significant important statistically. I mean, this is the autophagy 9, which is higher at the unpredictable and data restriction 40, but in males still it's not interesting to have, I mean, they don't want to change in the autophagy genesis still. This is autophagy 5, which is also similar in the unpredictable and in the data restriction 40, it shows higher on both of this one, but in males, the trend is still consistent, but still statistically it was not significant. We also see how sex-specific it. Yeah, the response to unpredictable, we will just focus on this one. Okay, at this level, they tend to become, I mean, to narrow the difference, but still the difference is on its position. While it is higher, the difference is higher here. So that means females were responding more intensively than the males for the daily unpredictability of diet. These are the sex-specific differences among, between males and females in different genes. We can see, we showed the, sorry for the similarity of the graphs, but this one is for female and this one is for male, if you are, if it is observable. So here, in the IGF-1 receptor, females showed high reduction, while in males, it's not that much. We can also see, especially this is in both unpredictable and data restriction 14. Here, the mTOR showed, and at the unpredictable one, the mTOR showed higher reduction, but not in the males. The males showed just reduction at the 40%, not at the, but the females, they show on both of the, so there is, we can indicate that there is sex-specific difference here. Here, there is also really, they are antagonistic. I don't know, this is this ribosomal protein kinase that we have to go in detail. It shows for females, initially it was low and then it increases and then it tended to decrease, but not significant. But in males, it was not affected by the unpredictable one, but on the data restriction 40, it increases. The initiation, eukaryotic initiation factor, okay, it was not statistically different, but still in females, it was higher. Here, in the ALK1 showed almost similar trend to this one, but females were initially low, but in males it increases a little bit similar, but increased at the 40%. That's all about the result. So let me just go into the final point, what has been found, what is the take-home message from our result. So we can conclude that gene expression mediates the effect of nutritional variability on the bird's phenotype. So we see the difference or the effect is almost similar, especially in the data restriction. The genes are affected, and the phenotype, the egg mass and the body mass was also affected, even the egg number. Birds shift nutritional budgeting between reproduction and self-maintenance depending on the severity of restriction. So when the nutrition is very severe, they tend to allocate much resource into the body mass, while the other one, when there is a moderate restriction, okay, they don't care about what happened in their body mass. They tend to allocate more resources into their reproduction. Individual variation in allocation strategy was related to mTOR. Yeah, we showed that in the result. Females showed intensified response to all treatments. We see females were really curious about this food. Yeah, we can say yes because they are laying eggs, but also even at the molecular level, they showed a difference. Catabolic genes showed consistently higher expression in males. The autophagy genes were consistently higher in males than in females. Okay, this can be one reason to help them to maintain their body mass, not just extremely changing or just reducing. So that is also very important. So in males, the catabolic genes, especially the autophagy genes, are still higher than the females. 2 weeks daily nutritional unpredictable did not affect reproduction and most of the gene expressionists. Yeah. The unpredictability, while we showed that in females there is a body mass reduction, but in terms of gene expression and reproduction, there was no as expected as we think that will be affected, especially in the reproduction. They don't show anything. So finally, I can say thank you for your attention, and I want to say thank you for my supervisors, which is also, we were just supported by Professor Chava in the animal science in formulating the nutrition and so on. So this is Professor Levente from animal science, for those of you who don't know him. Maybe, yeah. And these are our crew in the experiment and in the laboratory. Thank you, including Brigitta. She was always with us, and then the other guys are here. And those who are not here are also, I want to say, to convey my thanks to Debreceni University, and my university, undergrad university, is also I want to say thank you for helping me, and I say thank you for you guys for coming, and the discussion is open.
Thank you for this nice and resultful presentation. Now we have time for questions. Okay, Zoe.
Okay, I have a very nice talk, so I have a two-part question. One is that you found contradictory results. I think in your first experiment you found contradictory results in gene expression to what you expected. So I assume you didn't switch the samples, right? So you guys paid attention. So how do you explain these contradictory results, like in growth hormone expression and some of the other stuff? IGF-1, you expected, you said you found decrease, right? So
actually we have been trying to find what is the reason behind. Actually, we cannot even corroborate it with other results, previously studied results. We don't find even in the publication procedure we got this kind of questions, but still, the only thing we can say is it is similar on males and females. That gives us something, a message. Okay, what happened to the birds? Okay, for example, in growth hormone, Gene expression, growth hormone receptor is increased with the incoming growth hormone from the brain. So growth hormone is released from the brain, and it initiates the growth hormone receptor gene expression. Okay, sometimes, we can find it very relevant that growth hormone is most of the time higher in our blood during fasting, yes? When there is shortage of food, growth hormone is higher, it's released. That is against the insulin, where insulin is just produced, growth hormone will be increasing, so that will just initiate the activity or the expression of growth hormone receptor. Okay. Food by itself can also affect growth hormone receptor expression. So while we have less amount of resource, the growth hormone receptor is increasing its expression. That is for us still we cannot just say something at this stage. We have to go through. We think maybe another experiments, not maybe with me but with other our friends here. So. That's what I can say. Regard to the ribosomal protein kinase, that's also against our expectation. We were expecting that this gene is an anabolic gene. It will decrease with restriction, but it showed increasing result. Even we were just skeptical to say something about this, and we repeated in the unpredictable one. Even in the unpredictable one, it doesn't change its idea. It is continue to either show no effect or increasing, especially in the males. So we are saying that this gene can be expressed during lack of food or food limitation, but we know that having gene expression is totally different with the kinase activity. So we can have a lot of proteins that will be activated, but we can also have less protein. But when mTOR is active, it will activate it and it will be working. So the kinase activity may be different with the expression of these genes. So I think we need to focus on this one also. We open this even in our publications. We will see what we'll find in the future.
Okay, and the second part of my question. So you're studying resource allocation, and your assumption was that resources are scarce, right? But you're studying it in domesticated species that are selected for growth or selected for reproduction, reproductive output. So is this really something that can be generalized to other organisms? I know it's an evil question, but I'm just curious about your opinion.
Thank you very much. Yeah, of course, these species are domesticated species, so they have still domestic behavior. That's why we call the Japanese quail model birds, laboratory birds. So Japanese quail are model the laboratory birds for the domesticated chicken, and even people are saying something about the outside birds depending on the results found from Japanese quails. Yeah, we say that there is less resource outside. Okay, we try to imitate this by data restriction and to see how the molecular mechanisms are working. Actually, yeah, this is a worthy question, but still we can say we may not exactly say this is totally the same to all birds, but still there is we can have a bit generalization about what the behavior or the molecular content of birds. So due to this limitation in data availability, okay, we see this kind of gene expression results. So if people want to say something about the outside birds, yeah, this can be used as a baseline. Not really to totally generalize everything.
Maybe we have time for one more question. Okay, I have one. We could see some interesting results with males showing higher expressions at some gene expression levels. How do you interpret actually? Can we say that males are more susceptible to these physiological changes? Can we say generally?
Yeah, in males, not all genes are really higher in males. There are some genes, for example, these mTOR, these catabolic genes, most of the catabolic genes are higher in males, in females than in males. While the catabolic genes are higher in males. And okay, we can think that so males are utilizing their resources when there is less amount of resource. So when the catabolic genes are expressed, so they are tending to mobilize the resource inside the cell because of remobilization of the dead organelles, dead DNAs, and I mean this, DNAs and so on, the damaged DNAs inside the cell. This may help them to maintain their body mass. They are not tending to reduce their body mass as equal as these females. This can be related to that, but it needs still to make it more general. It needs another study. But I can say that males are not higher in all of the genes, but on the catabolic genes. On the anabolic genes, the genes in females are very high.
So if you don't have questions, then we can go to the mezzo together and continue the discussion. Thank you.